Good day, students. Um, Today we are going to treat yet another topic under enzymology, as advanced enzymology. That's BCH 401. And we are going to be looking at the topic multi-enzyme complexes. I am Dr. Obinna Uje. Now we are going to look at these multi-enzyme complexes and we are going to find out what are multi-enzyme complexes. And we are saying they are stable assembly of more than one enzyme generally involved in sequential catalytic transformation. In other words, we are saying that these are complex of many enzymes, but their interactions can catalyze probably one reaction or a series of reactions, okay? But they are existing as one entity. So, and in that entity or series of polypeptides, they have many enzymes attached to it. And that is why it's called multi-enzyme complexes. Now, what I'm trying to explain is that it contains several copies of several enzymes or polypeptide chain that is packed into one assembly. And they carry out a single or a series of biochemical reactions that are taking place in the cell. Now, let's see some examples of these multi-enzyme complexes. We have the first one we will look at is the pyruvate dehydrogenase complex. It's not just calling it pyruvate dehydrogenase, but recognize that it's a complex. So it is pyruvate dehydrogenase complex. Some people abbreviate it to be PDH, while others abbreviate it to be PDC, pyruvate dehydrogenase complex. You have another complex which we call fatty acid synthetase complex. You have yet another one, glutamine synthetase complex. You have the proteasomes complex, and you have the rubisco complex. Now, those are the examples that we'll consider. Now, let's dive in to look at this pyruvate dehydrogenase complex, which I designate as PDC. In the breakdown of glucose to generate energy, the glucose molecule is first broken down into two pyruvate molecules. One glucose molecule, through the process of glycolysis, is broken down into two pyruvate molecules. Now, this reaction takes place in the cytosol. It takes place in the cytosol. But pyruvate is converted to acetyl-CoA, which enters into the TCA cycle, is in the mitochondrial matrix. So, the pyruvate that you see in the cytosol must be transported from this cytosol into the mitochondrial matrix. And for it to do that, it means it must pass through the outer and the inner mitochondrial membrane. So the inner mitochondrial membrane contains a carrier or a protein that carries pyruvate across this membrane. So you have the mitochondrial pyruvate carrier will carry pyruvate from the cytosol to the inner matrix of the mitochondria. And then the pyruvate dehydrogenase complex will now catalyze the breakdown of pyruvate into acetyl-CoA. So let's look at these complexes and how and what happens. So now, all I have said is one, that pyruvate is oxidatively decarboxylated by this pyruvate dehydrogenase complex to form acetyl-CoA, and this takes place in the mitochondrial matrix. This is an irreversible reaction. So its irreversible reaction is the link between glycolysis and the citric acid cycle. And what is this reaction? Is that pyruvate, in the presence of coenzyme A and NAD+, will be catalyzed by the PDH complex to give you acetyl-CoA, CO2, and NADH. And we are saying that this pyruvate hydrogen complex is largely and highly integrated complex of 3 kinds of enzyme and 5 coenzymes. We are just saying that there are 3 kinds of enzymes, okay? And there are also 5 coenzymes. 5 coenzymes. So you must have this as in our basis. 3 different kinds of enzyme, 5 coenzymes. In other words, one of the enzyme will contain 2 coenzymes. We contain 1 enzyme, then the remaining 2 will contain 2 coenzymes, as we see in a jiffy. So, what are these enzymes and the coenzymes? Number 1, the 3 enzymes are designated E1, E2, E3. Now, E1 is known as pyruvate dehydrogenase, and the coenzyme that it contains is thiamine pyrophosphate. Thiamine pyrophosphate. And the place where do you get this thiamine pyrophosphate is from thiamine, which is vitamin B. The second one is E2, which is known as dihydrolipoyl transacetylase. Remember, the first one being a dehydrogenase means it is involved in an oxidation reduction reaction. Now, this second one is dihydrolipoyl transacetylase. And the coenzymes here is the lipoic acid and coenzyme A. They are generated or produced from vitamin B5, which is the pantothenic acid. So and then the third one, E3, is a dihydrolipoic dehydrogenase. Dihydrolipoyl dehydrogenase. And the coenzymes there is NAD and FAD. FAD is gotten from vitamin B, the riboflavin, and NAD plus is gotten from vitamin 3, which is niacin. Now, you will also note that thiamine pyrophosphate, lipoic acid, and FAD serves as catalytic cofactors, while coenzyme A and NAD plus are for stoichiometric factor. In other words, they are just there to balance the equation. Okay. Okay, let's continue. Now, let's look at how these conversion processes take place. The conversion of pyruvate into acetyl-CoA consists of 3 stages. One is the decarboxylation stage, you have the oxidation stage, and then the transfer of acetyl group to CoA. So if we look at this diagram, you find out that the pyruvate, which is a 3-carbon compound, loses CO2, and that is the decarboxylation process. Now, the acetyl group that is formed is oxidized, okay? And when it is oxidized, it enables the compound, the hydroxyl acetyl group, to be transferred to a CoH or coenzyme A, and then it will give you the acetyl CoA. So you see there is decarboxylation, there is oxidation, and there is transfer to coenzyme A to give you acetyl coenzyme A. Now, let's look at these 4 reactions. These 3 processes is carried out into 4 different steps. Like we said, the first step is decarboxylation, the second one is oxidation, the third one is the formation of acetyl-CoA, and the last one is the formation of NADH, which is a reducing component needed for the breakdown or to generate ATP. Now let's take this reactions step by step. Decarboxylation. This reaction is catalyzed by the E1. And remember that the cofactor of this coenzyme for this O1 is the thiamine pyrophosphate. So finally, the proteination yields um, hydro, hydroxyethyl thiamine pyrophosphate. So we are saying that pyruvate plus thiamine pyrophosphate, which is TPP, in the presence of a reducing agent, because addition of hydrogen is reduction, okay? In the presence of a reducing agent, we generate hydroxyl ethyl thiamine pyrophosphate and then give out CO2. So you can see where the ethyl group is attached to the hydroxyl ethyl TPP. Okay, after that you have an oxidation which is still catalyzed by the pyruvate dehydrogenase complex 1. And what happens is that the hydroxy ethyl group, which is attached to TPP, is oxidized to form acetyl group and transferred to lipoic amide. So the hydroxyl ethyl group plus lipomide, which this lipomide is in the oxidized form, okay? It will now transfer the acetyl group to the lipomide and then releasing TPP plus the acetyl lipomide. You can see the acetyl group is now attached, okay? The acetyl group, which is this group, is now attached in one of the S of the lipomide, okay? And this lipomide, which is in the oxidized form, is then reduced halfway. We say that it is now half reduced. Now, in the next reaction, which is the formation of acetyl-CoA, which is catalyzed by dihydrolipoyl transacetylase. As a transacetylase, it means that there is a transfer of an acetyl group. So we say the acetyl group is transferred from acetyl lipomide to CoA. And remember, when it transfers it to CoA, it forms acetyl-CoA, which fuels the um, citric acid cycle to generate ATP, and this acetyl-CoA is generated from pyruvate. So what we are seeing is that the acetyl lipomide plus CoAsh, which is coenzyme A, will give you acetyl-CoA plus dihydroxyl lipomide. Now you see that this dihydroxyl lipomide is now fully reduced. Okay, initially we said it was half reduced because there was an acetyl group that is attached to one of the S, but now the acetyl group had been transferred to coenzyme A to form acetyl coenzyme A and giving us the reduced form of the lipomide. The next thing is the formation of NADH. Now, the oxidized form of lipomide is generated by dihydrolipoic dehydrogenase, which is our E3. Now, 2 electrons are transferred to an FAD prosthetic group of the enzyme. In other words, the enzyme dihydrolipoic dehydrogenase contains a prosthetic group which is which acts as a coenzyme and then transfers it again to NAD+. Now, what we are just saying is that the electrons are transferred from this dihydrolipamide to FAD. That is just removing these 2 hydrogen, and you find out that it converts this reduced form of our lipomide or lipoate to the oxidized form, while FAD is converted to FADH2. Now, the FAG2 also quickly transfers the same electron to the NAD, and when it does that, it's going to form FAD NADH plus H. This electron transfer from FAD is unusual because the common role of SAD is to receive electrons from NADH. But this time around, it seems that it's doing the reverse. And this can only happen when FAD is acting as a prosthetic group. But when it is occurring as a molecule, it normally accepts an electron from NADH. Note that. So let's look at the overall reaction. In the overall reaction, acetyl-CoA is the main product of pyruvate dehydrogenase complex in aerobic respiration. Remember, cytosol, the reaction there is anaerobic, but in the mitochondria, the reaction there is aerobic. So this is just the overall reaction. Pyruvate plus CoA plus NAD plus will give you acetyl-CoA plus CO2 plus NAD plus plus hydrogen ion. Now, when you look at this, you'll find out that FAD didn't show up at all. Okay, that is why in the main reaction, you don't see everything. And because it's occurring as a complex, having many enzymes, we just write out the ones that are very, very important. So, and the process is very, for us to note again that pyruvate is that decarbonylation reaction occurs. You have your oxidation removing two electrons, and then you have the transfer of the carboxylic, the ethyl hydroethyl group to acetyl-CoA, and then the formation of the acetyl-CoA is done. Now let's look at the summary. What actually happens? Let's summarize these things. And this thing I'm going to show you, I want you to pay great attention because this is what you see in textbooks, and most of the times it gets confused. So what happens is that our pyruvate is converted by E1, pyruvate dehydrogenase. Now the E1 has a coenzyme PTT. Now this PTT will help it to lose CO2, and when it loses CO2, the PTT is converted to hydroxyl ethyl PTT. Okay, just look at it, it's still carrying the ethyl hydroxyl group. Now, what happens next is that the hydroxyl ethyl PTT will be converted to TPP. But how does it do that? It has to employ an oxidizing agent, which is the oxidized form of the lipoate. Okay? Now, and the enzyme that will help you do this is the E2. Now, E2 will now catalyze the transfer of the hydroxyl ethyl group to the oxidized form of the lipoate, and it will form an acetylated lipoate. And this acetylated lipoate, I'll tell you, I said that they are assumed or they are half oxidized. They are not half reduced. They are not fully reduced. So if you look at it very well, you will find out that the SH here is reduced, while the other S is not reduced. Okay, so what happens from here is that coenzyme A, or what we now normally write as CoAsh, will now receive the acetyl group or the hydroxyl acetyl group from this acetylated lipoate to form acetyl-CoA, which is the product we form. And then when it removes it, lipoweight is completely reduced now in the reduced state. And for this reaction to be going on, it means that we need, for instance, in this first place, we need to, the TPP we use, we need to regenerate it, okay? And the oxidized lipoids we use that is now reduced need to be re-oxidized. So how does it get re-oxidized? The enzyme 3 comes into play. And this enzyme 3 contains FAD. Are we following now? It contains SAD. And the FAD, we take the two hydrogen. There are two hydrogen attached to S. Each S has one hydrogen. It will take the two hydrogen in form of electrons and transfer it to FAD to form FADH2. Okay, while it returns to the oxidized form of the lipoids. Now, after that, the next thing that will happen is that we need to regenerate our FAD. And for us to do that, we will need NAD plus. And the enzyme that is still doing it is our E3. And when it does that, the FADH2 will transfer two hydrogen to NAD plus. And when it transfers the two hydrogen to NAD plus, we will now have NADH plus hydrogen ion. There is a plus in this second hydrogen. So when this happens, you find out that we have generated our NAD, okay, and also our acetyl-CoA. The acetyl-CoA will enter the citric acid cycle or the TCA cycle, and then energy generation completes. So in overall reaction, we are saying that pyruvate in the presence of CoH, pyruvate in the presence of the pyruvate dehydrogenase complex, which is E1 plus E2 plus E3, and in the presence of NAD, we give you NADH plus acetyl-CoA plus CO2. Please note this schematic diagram very well. So what are we saying in effect? I just decided to put this because most of us are afraid of the other one. We can now look at this. But the truth is, when you present this in a schematic, in the name of a schematic diagram, you will lose points. Okay? But they are just the same thing, where we are saying that pyruvate dehydrogenase converts, breaks down the pyruvate, releasing CO2 using the TPP to form the hydroxyethyl TPP. And then the next enzyme, E2, which is the dihydrolipoic transacetylate, carries, using the lipomide, oxidized form of the lipomide, will take the hydroxyl ethyl group, forming the acetylated and the acetylated lipomide, and then returning ATPP to its normal form. And when that happens again, CoH collects the acetyl group to form acetyl-CoA and the full reduced form of the lipomide. So the lipomide here is in the reduced form. That the E3, the E3, which is the dihydrolipoic dehydrogenase, will collect two electrons from the reduced form of the lipomide to form the oxidized form of lipomide again, and thereby transferring that electron to FAD, and you have SADH2. Now, SADH2, in the presence of NAD plus, we now collect these two electrons to return to FAD, and then transfer the electrons to NAD plus and then hydrogen plus, which NAD plus can now enter the electron transport chain or even these to generate 2.5 ATP. And that is a story for another day. Now, what is just the significance of this reaction? Number one, we say that the reaction is completely irreversible. Two, it is the committed step towards the oxidation of glucose. And number three here is that in this process, there is no net synthesis of glucose from fat. Okay, so the reaction we are talking about, fat does not contribute to it. It is purely pyruvate, and this pyruvate is coming directly from the oxidation of glucose molecule. So how do we regulate pyruvate dehydrogenase? To regulate pyruvate dehydrogenase, to regulate pyruvate dehydrogenase, so let's look at the regulations. Let's look at the regulations. Um, I just hope this. And in the regulation, remember that the essence of pyruvate going to acetyl-CoA in the first place is to generate ATP. Now, when ATP has been generated, there is acetyl-CoA, there is NADH, the reduced form of NADH. When they are present, it then means that this complex needs to be shut down. And how do they shut down this or inactivate this pyruvate dehydrogenase complex? Is by activating another enzyme. And this enzyme is known as pyruvate dehydrogenase kinase, pyruvate dehydrogenase kinase. Now, as a kinase, remember the functions of kinases is to phosphorylate, okay? So when pyruvate dehydrogenase complex is being phosphorylated, it will be deactivated. And when it is deactivated, there will not be any need for it to produce acetyl-CoA or energy. Okay? Then on the other hand, when the concentration of pyruvate is high, ADP is high, coenzyme A is available, NADH is there. There will be need for ATP to be produced. And what will happen is that this present will go and inhibit NADH kinase. And when the kinase is inhibited, then pyruvate can go ahead because nothing will be there to phosphorylate the PDH. Now, but there is another case where the body needs to generate energy. For instance, the heart that is continually to beat, whenever the muscle contracts, they can release calcium ion. Okay, the heart needs energy all the time. So even when there is phosphorylation, the body brings up a mechanism in which it will allow these pyruvates to form acetyl-CoA. So when the muscle contracts, it releases calcium, and calcium goes to activate PDH phosphatase. PDH phosphatase. Okay? And what will phosphatases do? They will go to dephosphorylate, remove the phosphate group and then allow pyruvate to go to acetyl-CoA. So I will stop here, or rather, let me continue. I think I have only one more slide to go. What are the significance of PDC? Number one, the clinical significance. There are cases where you have pyruvate dehydrogenase deficiency, and that is where E1 is deficient. What in this place, what happens is that there is an accumulation of pyruvate, and then this pyruvate will kind of, instead of forming acetyl-CoA, we form lactic acid. And then that is what is responsible for lactic acid acidosis. Okay? And we say it is the inability of the pyruvate to be converted to acetyl-CoA because pyruvate dehydrogenase is not really available. And when this happens in the brain, the acidic level in the brain increases, leading to neurodegeneration. And then sometimes, even in children, it can lead to death. Now, how do you handle it? Sometimes you handle it through restriction of carbohydrate diets. You restrict the person from eating a lot of carbohydrate diets, and then also supplement with thiamine or thiamine pyrophosphate, so that even so it since it's deficient, the little quantity that you have of the enzyme we have enough coenzymes to work with. Another clinical significance is in the issue of arsenic poisoning. And then, and in arsenic poisoning, the lipoic acid, okay, is the one that is being hindered. And when that lipoic acid is not there to actually reduce, to carry the acetyl group to coenzyme A, then it makes also the accumulation of pyruvate, cause also the accumulation of pyruvate. So pyruvate, like we have already said, once it accumulates in the brain, we already know that the best available option for the body is for it to be converted to lactic acid. And we know that there are a lot of brain issues, neurological conditions, and even death could occur. So I want to thank you for this lecture, and I will ask you to make sure you do your assignments, and that's all.